Assalamu alaikum everyone. My name is Hasib Asif. I'm the group lead for the group, which is now be presenting on the design of non-invasive blood urea and creatinine meter, which is prepared by Hasib Asif, myself, Daniyan Hussain, Aram Siddiqui, Sayyid Ammaruddin, and Ahmad Aliyan. Our problem statement is basically that the patients with urea and creatinine imbalance, they are actually prone to renal disorders, which can cause severe health issues, especially if patient is on dialysis. To manage kidney health in such patients, they need to observe their urea and creatinine level in blood regularly, which is being done invasively up till now by doing venipuncture, which is quite painful for the patient, as well as they are prone to infections, which is and that is quite costly for patients if they do it regularly. And you all would know that such patients need to do this regularly. They do not have any other choice. Now, what is urea and creatinine actually? Urea and creatinine basically are nitrogenous end products of metabolism. They are the primary metabolite derived from the dietary protein and tissue protein turnover. Creatinine is the product of muscle creatinine catabolism. They are both relatively quite small, 60 to 113 Daltons. This is basically the measuring unit. They are distributed throughout the body water. Now, what are the problems and diseases that are caused by abnormal urea and creatinine level? Number one, hypercreatinine level problems. Number two, hypocreatinine level problems. Number three, liver diseases. And number four, the muscle diseases. Now, why urea and creatinine monitoring is very important? This is because they need to ensure the glomerular filtration rate. The reasoning for the utilization of creatinine or urea estimation is to basically evaluate the renal capacity in that, and that is the plasma and serum levels of both reflect the glomerular filtration rate, the boundary that characterizes kidney work for the clinician. Acute pancreatitis. This is the estimation of serum plasma urea, and they have an incentive in the early trauma center. Appraisal of patients with intense pancreatitis, and number three is hemodialysis. So the objective of this project is basically to provide a non-invasive approach to measure and monitor creatinine and urea in blood. Contrary to the current invasive and painful methods which are now being used using the venipuncture, our device would be convenient as well as it would be cheap, and it would not also cause the contamination, which is so far the biggest challenge for the patients. To develop such a non-invasive urea and creatinine concentration, the measurement instrumentation device, the infrared light and its absorption depending on various urea and creatinine concentrations will be examined. So why non-invasive technique is better? Because it utilizes the sensors, number one. There is no skin piercing whatsoever. There is no contamination and blood loss. As you would know that when we do the venipuncture thing, there is contamination and there is a loss of blood as well. It is quite painless. It prevents the infection because there is no need for the any other needle or that kind of a thing to enter into our skin. And number four, it is accurate as compared to the traditional way of checking the urea and creatinine level. Now, this is actually the comparison of invasive and non-invasive technology. You can also say that this is the value creation which we are going to offer from our device. So number one, if we see the invasive technology, this is basically quite painful because pricking is involved. It requires blood. It is unsuitable for hemophobia. It is periodic buying of test strips that is also required. It is less accurate, and many infections and viruses may grow because of it. Whereas if you see the non-invasive technology which we are going to offer, it is going to be painless. There is no blood requirement. It would be suitable for hemophobia, and no periodic buying of test strips, which is currently being used in the traditional way of checking the creatinine and urea level. It would be quite accurate. It would be infection free, and the cost will be only the initial purchasing cost. That's it. no use of alcohol or antiseptic in our technique. So what are the sites which we'll be using in the body for non-invasive monitoring? Number one, nasal septum. Number two, earlobe. Number three, through the finger bed. Number four, through interstitial fluids from sweat gland. Now, what are the possible non-invasive techniques? Number one, infrared spectroscopy. What is infrared spectroscopy? It is the analysis of infrared light interacting with the molecule. This can be analyzed in three ways: by measuring absorption, emission, and reflection. The main use of this technique is in organic and inorganic chemistry. Reverse and anthophoresis, where anthophoresis is the use of electrical currents to deliver drugs through the skin using electric currents. Reverse anthophoresis takes molecules out of the skin using electric currents. This is basically the block diagram of our device. There'll be a battery, there'll be a voltage regulator, an LCD, an amplifier, analog to digital computer, and the other things. So what components are we going to use? Number one, photo detector will be used. Number two, the voltage regulator. Number three would be the operational amplifier. Number four will be the display so that the readings can be seen. And choice of wavelength. This is quite important because the wavelength actually shows you the reading. Short wavelength near infrared 640 to 1000 nm spectra of aqueous solution of urea has been monitored. The observation yields that maximum absorption occurs in the range of 920 to 950 nm. So the selected wavelength which we have selected for our device is basically the 940 nm for non-invasive urea meter. This is actually the wavelength which we'll be using in our device. So this is our signal flow chart, how the signals are going to travel in our device. This is the circuit diagram. This is basically the business model canvas. Our key partners are going to be the suppliers from foreign countries, especially China, because it is not these devices or the components are not available locally. The second thing is the key activities. The activities would include the designing and fabrication, manufacturing of this device, the quality checks, and the clinical trials. Key resources would be capital, electronic simulation, softwares, electronic components, and engineers. Value proposition is the most important thing, and that is going to be the convenience we are going to provide through our device. There will be no contamination whatsoever. It would be quite easier and accessible. You do not have to go to the doctors every day. It would be quite cost effective because the initial cost, there will be an initial cost of the purchasing of device, and that's it. It would be quite painless, and the output would be immediate, because when you do the venipuncture thing, you need a certain bit of time to get the results. It would be alcohol and antiseptic free. The customer's relationship segment is also quite relevant, because how we are going to manage our relationships with the customer. It is going to be the self-service kind of a thing, because everyone can do it on their own. There'll be our device is automated. It would be non-invasive and the low cost. These are the things which are going to obviously attract the customers when they are going to know about it. The channels we'll be using are the doctors, the laboratories, the hospitals, and the pharmacy, because obviously it is a medical equipment. The customer segments which we are going to target is every individual, firstly, because obviously no matter you're the patient or not, it is always good to keep a check on your health, and so that's where every individual is our customer. We'll be targeting the hospitals, pharmacies, and wholesalers. So the cost structure is basically going to be the electronic circuitry, which will be used in the device, the fabrication of probe and device, the human resource which we are going to require, the IT resource, the technical staff. Revenue streams are going to be the hospitals, general public, pharmacies, wholesalers, because we are going to sell them. So the softwares which we used for our simulation were Proteus 8, Arduino IDE, Arduino library. This is a video which we made. Concept of our device this is another video. So the conclusion is that majority of the devices currently which are used to monitor the blood and urea and creatinine level, they are not automated at all. We are still using the traditional way of doing things, and therefore the objective of our project was to design a device which would fill these gaps in the market, which would target the customers who are actually fed up of, you know, doing or consuming the traditional way of doing things. Our device would also ensure that there is no blood loss or contamination. The removal of cons of non-invasive methodology would improve the blood urea and creatinine level monitoring, because everyone's gonna find it easier to do such kind of a things. A device would also help in the prevention and treatment of common diseases, because if we are going to track your urea and creatinine level on the daily basis, it would be quite good for you to stay away from the diseases or harmful, things which could happen to one. If one can develop this circuitry physically, it can be targeted to hospitals, diagnostic centers, and household patients. So our recommendations are that this device should be physically tested first. We have only done the research work for urea detection, although we were supposed to design the device for creatinine detection as well, but due to the shortage of time and resources, we are not able to do so. Our group designed this device for urea detection, whereas similar devices can be used for BUN as well, which is blood urea nitration. So there is a lot of work which can be done in this segment. So this should be done. This is what our recommendation is. So this is the end of our presentation. If you have any questions, you can ask. That's it from my side and from my group. Thank you so much. My name is Asif Asif, signing off.